Hello class, and welcome back to part 2 of your brain and cranial nerves lecture. We're going to talk a little bit about the information processing that occurs within the brain. The cerebral cortex receives input from sensory organs as well as the somatosensory receptors. These are specific types of sensory input which enter the primary sensory areas of the brain lobes. There are adjacent areas that process features and sensory input and integrate information from different sensory areas. The somasensory and motor cortexes, neurons are distributed according to the body part that generate the sensory input or receive motor input. So both the frontal lobe and the parietal lobe have two cortexes, as we've said, that separate them. And so we have the motor cortex as well as the somasensory cortex. And you can see that the motor cortex ends up taking control of different areas of the body and their motor control. Sensory cortex, however, is required in processing of sensory input. Studies of the brain have mapped areas responsible for language and speech. Broca's area, this is in the frontal lobe, and it is activated when speech is generated. The Wernicke's area resides in the temporal lobe, and this is active when speech is heard. There's also a FOXP2 gene that deals with language, but we won't get into that. So, emotions. These are generated and experienced by the limbic system and other parts of the brain, including the sensory areas. The limbic system is a ring of structures around the brainstem and include the hippocampus, parts of the thalamus, amygdala, which is in the temporal lobe, and helps to store emotional experiences as emotional memories. So, memory development and learning. There are two processes that dominate the embryonic nervous system development. Neurons compete for growth supporting factors in order to survive, as only half of the synapses that form during embryo development survive into adulthood. So, the learning, when neurons make new connections or when strength of existing neural connections change, this is called a neuroplasticity. Neuroplasticity describes the ability of the nervous system to be modified after birth. This changes can strengthen or weaken signals at the synapses depending on what's going on. If you're trying to learn something or if you have dementia. Those are just two examples. So there are two types of memory. Your short-term memory is accessed via the hippocampus. Long-term memory, however, the hippocampus does help form and then stores it in the cerebral cortex. So, the central nervous system. Okay, so we're going to go over the cranial nerves just a little bit. These all originate within the brain, and most of them terminate in the organs of the head and upper body. So you will see your olfactory, one. These are the olfactory bulbs. The oculomotor, which is right here. Optic. Your optic chiasm is right here. Truncular, abducent, vestibular coccular, hypoglossal, accessory, your vagus, Glossopharyngeal, facial, trigeminal. Okay, and you will notice that each of these are numbered. That will be very important on your practical and most likely exam for the lecture section. This table just goes off of what exactly each of the cranial nerves does and the type that it is, because it's either sensory or primary motor, and some of them are mixed. This just is an easier photo to be able to look at. It's a good one that shows not only the photos of it, but also tells you what it is and where it goes. The purpose of sheep brain dissections is kind of threefold. You're going to familiarize yourself with the 3D structure of the brain. We're not going to be doing these in class, so you're going to have to kind of pretend along with me. You do have a Connect version, so we'll see how that goes. You're going to locate structures and understand how structures relate to each other. Identifying structures and reflect on how their relative positions dictate their functions in the brain, or anatomy precedes physiology. This is the front of the brain. You will notice that over here is the brain stem, and then you have the anterior or rostral, medial, and lateral. If we turn it on its side, you have the ventral down at the bottom, and the dorsal on top, again the rostral, and the caudal. In animals, the posterior side is called caudal. You'll notice this when it comes next semester for the pigs. So you have a frontal or coronal plane, and I will end up cutting a brain that way, as well as the horizontal. I'm going to do a coronal cut as well as a sagittal on mine. So again, this is the cuts that will be made right across here, coronal, and down the middle is the sagittal. Okay, you will also notice that the bumps are on the outside, and those are gyruses. Remember, you gyrate with the gyruses, and the sulcus are the depressions. They're sulking. So, for your practical exam, if I am going to ask you about the lobes, I will put pins in different places, but you will most definitely know exactly where it is. So if I put it here, it's the optical. If I put it over here on the side, temporal, parietal, and frontal would be all the way over here. This is called the longitudinal fissure, and a very good possibility that I would put a pin right there. So if you turn it over, most of the time, depending on how the dissection has gone, you will notice the olfactory bulbs, the optic nerve, which is coming out right here, the optic chiasm, the pituitary gland, which kind of hangs down just a little bit. And then you have these two nodules right here, and those are the pons. This bigger lump is the medulla oblongata, and then you have the spinal cord. And so, once a mid-sagittal section has been established, you have the cerebral hemisphere, and this beautiful white matter is the arbor vitae. Of course, the spinal cord, the medulla oblongata is the first bump, and then you have the pons. And see, the pituitary gland just kind of hangs down. Optic chiasm, the hypothalamus, which kind of goes like that, and the thalamus, the corpus callosum is this white portion right here. And of course you have the frontal lobe, the olfactory bulb, the parietal lobe, the occipital lobe, and the pineal body. Okay. So again, you're looking at the different ventricles right in here. And if you go past, this is not the best photo, but you see that there is a membrane. That's the septum reticulum. And if you go under it, that is your lateral ventricle. So a coronal section is really good to be able to see the ventricles. You can definitely see the horns here, which are the ventricles. These are the lateral. And then you have the third. So, and this right here is the hippocampus, and that is located directly under the lateral ventricle. You can see the corpus callosum, and this is what is binding together both the left and the right side. And this is another corpus callosum, and you can definitely see on this photo how beautifully white it is. The hippocampus again, as well as the lateral ventricle and the thymus. Okay, so this is the end of the second video for the brain and nervous system. I will be going to the university, and you will find a video and that goes over the dissection just a little bit more. And you can also, of course, look at YouTube for different videos. If you have any questions or comments, please let me know. I will try to use either extraordinarily good photos or something exactly like you would see in the lab for your practical. Have a great week, and I will see you again soon. Goodbye.